Mutations in Tubb8 and human oocyte meiotic arrest. Human reproduction depends on the fusion of a mature oocyte with a sperm cell to form a fertilized egg. The genetic events that lead to the arrest of human oocyte maturation are unknown.
Oogenesis is the process by which the female gametes, or ova, are created. The female gamete is called an ovum. Sometimes people will refer to female gametes as eggs, but the term egg can include more than one stage of development, and the definition of an egg also changes depending on the type of organism. For example, the entire prenatal development of birds occurs inside an egg, but in placental mammals, after the egg is fertilized and starts dividing, nobody calls it an egg anymore. So, we're going to use the more precise term for a female gamete, which is ovum, or ova for the plural form. Now you may recall, each ovum must be haploid and contain only one copy of each chromosome. You may also remember that in order to create haploid gametes, a cell must go through the process of meiosis, which involves replicating its genome and then dividing, not once, but twice. In theory, this would create 4 haploid gametes from a single diploid cell, like it does during spermatogenesis. However, this isn't the case for human female gametes. Let's go through the process of oogenesis to see how this works and how many haploid gametes are made from a diploid cell. The diploid germ cells that have the potential to develop into ova are called oogonia. In humans, all of a female's oogonia that she will make in her lifetime are created when she's still a fetus and hasn't even been born yet. In fact, about 1 or 2 months before a baby girl is born, most of her approximately 7 million oogonia die, and the remaining surviving oogonia enter meiosis I and become primary oocytes. These primary oocytes press the pause button on their development in prophase I, after they've replicated their genomes, but before they've made the first meiotic division. They stay arrested at this stage of development for over a decade until the girl begins her first menstrual cycle. Then, for about the next 30 to 45 years, on a monthly basis, primary oocytes resume meiosis where they left off and complete the first meiotic division. When the primary oocyte does finally complete its first meiotic division, it divides the chromosomes evenly, just as you would expect. However, it does not divide its cytoplasm equally. Almost all of the cytoplasm remains in one of the 2 daughter cells, which becomes a secondary oocyte. The other daughter cell, which gets half of the chromosomes but very little cytoplasm, is called a polar body. The polar body is not a functional oocyte. Instead, it degenerates and dies. The formation of a polar body allows the primary oocyte to reduce its genome by half and conserve most of its cytoplasm in the secondary oocyte. The secondary oocyte still has 2 copies of each chromosome, so if it's going to become a fully functional ovum, it must undergo the second meiotic division. This division is also uneven like the first one, with half of the chromosomes going to another very small degenerate polar body, and half of the chromosomes being retained by the ovum, along with almost all of the cytoplasm. In this way, the ovum achieves its haploid state while conserving as much cytoplasm as possible. In humans, oocyte development occurs in the ovaries. Each oocyte is surrounded by a number of follicle cells to create a follicle. When the menstrual cycle begins, one or maybe even a few primary oocytes begin to grow larger, and the follicle cells increase in number and cause the follicle to grow larger too. Usually, some of the developing oocytes will degenerate and only leave one follicle that will mature, but every once in a while, 2 or even more follicles will mature. In these cases, fraternal twins or triplets that are genetically distinct may be born. In any case, as a follicle reaches maturity, the primary oocyte completes its first meiotic division and becomes a secondary oocyte. Very soon after that, the follicle ruptures and the secondary oocyte is released into the fallopian or uterine tube, even though the second meiotic division hasn't occurred yet. The release of a secondary oocyte from the ovaries is called ovulation. Once released from the ovary, the oocyte slowly travels down the fallopian tube toward the uterus. Fertilization usually occurs in the part of the fallopian tube nearest the ovary. This is because the oocyte is only viable for up to 24 hours after ovulation. If it isn't fertilized by a sperm within this time period, then the oocyte never completes the second meiotic division, and so it never technically becomes an ovum. However, if the secondary oocyte is fertilized by a sperm, it immediately finishes its second meiotic division, releases a polar body to leave only a haploid female genome in the ovum, and then becomes a zygote as the nuclei of the sperm and egg fuse together. In summary, oogenesis is the process by which the female gametes, or ova, are created. The female gamete is called an ovum. The diploid germ cells that have the potential to develop into ova are called oogonia. In humans, all of a female's oogonia that she will make in her lifetime are created when she's still a fetus and hasn't even been born yet. By the time a baby girl is born, all of her oogonia have either entered meiosis I and become primary oocytes, or degenerated and died. The primary oocytes press the pause button on their development in prophase I, after they've replicated their genomes, but before they've made the first meiotic division. In humans, oocyte development occurs in the ovaries. Each oocyte is surrounded by a number of follicle cells to create a follicle. When the menstrual cycle begins, one or maybe even a few primary oocytes begin to grow larger, and the follicle cells increase in number and cause the follicle to grow larger too. Usually, only one follicle reaches maturity, and the primary oocyte in the follicle completes its first meiotic division and becomes a secondary oocyte. Very soon after that, the follicle ruptures, and the secondary oocyte is released into the fallopian or uterine tube before the second meiotic division has occurred. The release of a secondary oocyte from the ovaries is called ovulation. Once released from the ovary, the oocyte slowly travels down the fallopian tube toward the uterus. If the secondary oocyte is fertilized by a sperm, it immediately finishes its second meiotic division, releases a polar body to leave only a haploid female genome in the ovum, and then becomes a zygote as the nuclei of the sperm and ovum fuse together.
The expression of Tubb8 and all other tubulin isotypes was assessed in human oocytes, early embryos, sperm cells, and several somatic tissues by means of a quantitative reverse transcriptase polymerase chain reaction assay. It was sequenced the exams of 5 members of a 4-generation family, 3 of whom had infertility due to oocyte meiosis I arrest. It was performed same-year sequencing of a candidate gene, Tubb8, in DNA samples from these members, additionally family members, and members of 23 other affected families.
I'm Fu Choutang from Peking University. There are more than 200 types of cells within a human body. Probably one of the most important types of them is human eggs, because they are the only type of cells that can transmit all the genes from a mother to her children and keep the human species immortal. The human eggs are also very special in the way that every egg is genetically different. In fact, this is why every one of us is genetically unique except identical twins. Every egg cell is genetically different, so you have to analyze them one by one to really understand their unique genetics. We recently published a paper in Cell to show that by using cutting-edge technology of mobile single-cell genome amplification and deep sequencing, we can get a better understanding of these mysterious human eggs. Firstly, in a human egg, the two sets of genes coming from grandmother and grandfather need to be rearranged and mixed properly, similar to shuffle of pokers. Every of the gene strings called chromosomes need to be exchanged with its sister chromosome. The rearrangement in eggs is more thorough than that in sperm. We now have a better idea why that happens. Secondly, for the first time, we phase a woman's genome by sequencing her eggs. There are two sets of genes in every cell of a person. It's difficult to separate them to two distinct groups accurately. Now, taking the advantage that within a byproduct cell of an egg, called a second polar body, there is only one set, but not two sets of genes. We can easily separate the two sets of genes of a woman into two distinct groups. In this way, we can get a better understanding of the genetics of the woman.
I'm Dr. Tiao from Peking University Third Hospital. In vitro fertilization is a powerful technology to help infertile couples to get their own babies. The first IVF baby in the world was born in 1978 in the UK, and the first one in China was born in 1988 in our hospital. Although the IVF technology has been successfully used for more than 35 years, the successful rate is still relatively lower, around 30 to 40%. One of the main problems comes from genetic abnormality of human eggs called aneuploidy. It has been shown that when a woman is older than 35 years old, more than 30% of her eggs are genetically abnormal as aneuploidy. These abnormal eggs will lead to either miscarriage or birth of the baby with genetic disease. The real problem is that there is no accurate, efficient, and lower-cost way to detect them. Because of the genetic material within an egg is so tiny that it's too difficult to detect this abnormality with standard molecular biology tools. Fish, SNP, and CGH array have been developed successfully but with limited resolution and accuracy. We analyzed the two polar bodies of one human egg by deep sequencing, simply by counting the number of chromosomes in this byproduct cell as 0, 1, 2, 3, 4. We will know that if the egg is a normal one with two complete sets of genes, or an abnormal one lost of again some copies of genes. More than this, we can also screen gene mutation in these eggs inherited from the mother. If the mother has some gene copies out of work, we will know that if their out-of-work gene copies are transmitted into these eggs or not. In this way, we kill two birds with one stone. One deep sequencing analysis to avoid two types of genetic problems. We really hope to both help infertile couples to get their own babies in a much more efficient way and also reduce the extra pain of these families by avoiding the transmission of genetic disease from the mother to the baby.
Dependent bias. Some genes are amplified significantly more than others. Multiple displacement amplification (MDA), invented 12 years ago, offers better coverage, but still limited by the sequence-dependent bias. Our new whole genome amplification method, multiple annealing and bettering of life at a single molecule level.
Pros and cons. Pros. It was